sit down. Welcome back, everyone. So 2 more talks. I think they're going to be very exciting, and then we'll have a reception and a poster session. So our first talk is by Jean Yeo from UCSD, and he'll be talking about modulating RNA diseases using CRISPR-Cas9.
Great. Can you hear me at the back? OK. Thank you for the invitation to come here. I've not been here before. It's an amazing view of the building, and thanks for the fantastic talks this morning. So what I thought I'll do is not talk about stem cells, sorry, and not talk about genome editing. I apologize. Instead, I'm going to talk about a different application for CRISPR-Cas that we are excited about. And I think we'll come back eventually to our interest in actually using stem cells. We do a lot of stem cell work to generate ASD models, ALS models in the dish, but I'm not going to talk about that today. OK, so RNA, for folks that, oops, this is not moving. All right, I'll use this instead. OK, for those who have forgotten, we know RNA can be used to measure the identity of cells. You can use single-cell RNA-seq here frequently to do that. But we think of RNA as a more dynamic state where we have to, we have to understand how RNA is processed before the RNA molecule is then matured, exported to the cytoplasm, and then translated into proteins. And so this is a picture showing RNA is generated from, obviously, DNA, right, during transcription. And a lot of things happen here. So just to remind everybody what happens there, a lot of stuff, right? So you have things like splicing, polyadenylation, RNA modifications like methylation, pseudouracilation. There are more modifications on RNA than there are on DNA, transport, localization, translation, and so on. So a lot of things happen here. And over the last decade, we've spent a lot of time trying to study how RNAs get modulated by studying RNA binding proteins that modulate these things. But we also know that, that when you have defects in any of these processes or defects in interaction sites between RNA binding proteins and RNA, you can get many different diseases. And these are RNA-related diseases as follows. And I'll mention a little bit about myotonic dystrophy today and ALS. So these are diseases that have a strong RNA basis. OK, so over the last couple of years, we've started to switch in not just understanding how RNA is processed by studying what RNA binding proteins do, but by trying to engineer new ways to modulate these processes with sort of engineered approaches. So in the past, we had engineered RNA binding proteins that did this. But then we realized that you can also repurpose CRISPR-Cas proteins to bind and modulate RNA. And initially, we just wanted to study RNA processing, right? You can use these engineered proteins or engineered CRISPR-Cas proteins to modulate things like splicing, to methylate RNA, to do different things. So that was sort of the hope. But as the first pass, we wanted to see this even worked. And so we focused a little bit on localization. But then we realized we can do more with it. OK, so why RNA? Thinking about therapeutics, right? You've already probably seen the slide from many different talks in the CRISPR arena. There are obviously a lot of effort in trying to use CRISPR genome editing to make ex vivo sort of therapeutics, right? So you can modify cells, put them back in an individual. It's like SVD, for example, SCD. However, in vivo applications are still a bit difficult. And so we started thinking about what are the limitations. It's an amazing technology, but there are some limitations. I think there are still the risk of off-target edits. All you need is like one cell, maybe in a billion cells, to have an off-target edit, and that might be bad, maybe. Works really well in dividing cells, not so much in vivo and non-dividing cells. And for very highly polyploid organisms, where you have to modify things, modify genome, genetic material across many copies, that might be very difficult. We've been interested in viral RNAs, where they're not, maybe don't have a RT intermediate for DNA. You really have to edit only RNA, modify RNA. So we said, well, maybe we can try something different. And so we said, what about using the same systems but avoiding DNA? Try by just modifying RNA anyways. that then prevents the expression of bad toxic RNA or bad sort of toxic proteins, right? So just a reminder that we can obviously have 3 billion bases here, where if you have an off-target edit, that's sort of bad as permanent. But a few percent of the genome is expressed as protein coding genes, right? So you have a smaller window of like off-target edits, and worst case, it's sort of temporary, right? The RNA just generated, you made a mistake, it's destroyed, and it goes on. Okay, so we first came about this idea around 2013, 2014. And so this is a paper from Jennifer Doudna's lab showing the first repurposing of Cas9. So what she did was she took a dead Cas9 and then tricked it by putting a PAM oligomer. So you have an oligomer with a PAM recognition site in the oligo, and then you can trick Cas9 to bind in vitro to RNA. Okay, so this was sort of a demonstration in vitro, and then we worked with her and said, well, we are interested in RNA localization. Initially, it was in neurons, and so we said, well, just get a proof of concept. Can you then modify the PAM oligomer so it works in vivo? Let's trick the Cas9 protein to bind RNA by having the PAM oligomer hybridize to the RNA, have the guide RNA also then be designed to hybridize to the target mRNA, and then just fuse dead Cas9 with GFP. All right, so the idea here is that can you see an mRNA that you target move to the cytoplasm, and when it's there, it will localize most of the Cas9 GFP into the cytoplasm. So this is the sort of negative control, right, where you have Cas9 GFP stuck in the nucleus, right, because we stuck several nuclear localization signals on Cas9. But when you use a guide RNA and PAMR targeting GAPDH mRNA, you can see the signal is pretty clean. It sort of moves out into the cytoplasm. So demonstrated the first sort of way of bringing Cas to a sort of in vivo RNA application. And so we were actually rather embarrassed because right after this, there are a lot of people said, oh, you can do many things like treating muscular dystrophy, neurodegeneration, and so on, when in fact we hadn't really thought about this yet. But we figured since they mentioned it, why don't we work on it, right? And so we had a postdoc join the lab that was interested in myotonic dystrophy. And this is a terrible disease, right? So you have atrophy of muscle. There's also a CNS disorder where you have memory loss, sleep abnormalities, and cardiac conduction issues. In congenital cases, you have large expansions, which I'll mention about in the next slide. These kids suffer very quickly, and they're not usually as a fatal disease. So DM was our prototype model to think about targeting RNA-related diseases. Okay, it belongs to a class of diseases known as microsatellite expansion diseases. There are many of these, 30 to 40 of these diseases. And so they're typified by having DNA repeats, short ones, GAA or G4, C2, or in our case of DM, CTG repeats, which are expanded in a gene loci. And they can be expanded 1,000 copies of this short repeat, and they can be also sometimes heterogeneous within the sequence itself, right? And so, for example, in the case of DM1 and DM2 in muscular dystrophy type 1 and type 2, these repeats are simple, CTG repeats. They are expressed, right, in the 3' UTR of the DMPK gene and a different gene for DM2. So they're expressed, they're processed. In this case, it's an intron, in this case, it's a UTR. And what happens is they act as sinks. So they sequester proteins that like to bind them. For example, muscle-blind like 1, 2, and 3, which are RNA binding proteins that like to bind primary CTG sequences, but also structure, and they sequester them. So because these proteins are sequestered in the nucleus, then they cannot perform their normal functions, which is to control splicing of many downstream genes, so hundreds of genes. Okay, so you've got these repeats are expressed as RNA. They sequester proteins. And these repeats are abundant enough that you can do an RNA fish, and you can see them in the nucleus of these cells, right? Okay, so you disrupt splicing. It's bad. You get myotonic dystrophy. So we figured, can you first visualize, like we did before in our previous paper, can we visualize the repeats, right, with our Cas9? And so here is just GFP fused Cas9, right? Dead Cas9, and this is just RNA fish showing the with CAG, which is complementary to CTG. So you can see the CUG repeats in the cell. But when you treat the cells with targeting guide to CTG, and in this case also a targeting PAMR, then you can see pretty nice localization of Cas with the repeats. So Cas GFP is now binding to these repeats. And they're so highly abundant and repetitive that they start sequestering Cas9 to itself, right, to the repeat itself. But interestingly enough, when you don't even use the PAM oligomer, you can actually still recognize the foci. Maybe not as sensitive, but because there's so many repeats, they still sequester it without the PAM oligomer. So that means just Cas9 alone can just bind RNA. Turns out that this is true now from many labs. People have shown SA-Cas9, CJE Cas9 many. Also, orthologs of just Cas, S pyogenes Cas9 can also bind RNA. And so we were able to now show this with many other repetitive sequences. So the CAG repeat sequence in SCA1, 2, and Huntington's disease, DM2, CCUG repeats, right? So you can see localization with the repeats. Okay, so then we said, well, this is cool. We can see the repeats now in live cells. You can image them moving around. I'm not going to show any videos. But what was interesting was that now you can fuse these basically RNA binding protein that you just engineered with endonucleases. And so we can search for different endonucleases. We ended up using the PIN1 endonuclease from the SMUG, one of the SMUG orthologs, which is a low turnover enzyme. So that means that you can recruit Cas to these repeats and chew it up, right? So you can destroy the repeats. And so these are cells in green that express Cas9 with the targeting guide. And you can see that cells which express it, destroy, have no more repeats, right? So the repeats are gone. So you can show this with northern blots. You can show this with qPCR of the allele with the repeat, for example. And you can show that this works for many different repeats in different diseases, ALS, DM1, DM2, and so on. So one question was, since it's Cas9, how do you know it's just not binding DNA as well as just RNA, right? And so we did this experiment where we used a doxycycline inducible system, TET inducible system, where on the same promoter here, on the same TET response element here, you can express GFP on one side, and then on the other side, you express the DMPK locus with the CTG repeats. Okay, so the experiment was as follows. We wanted to show that Cas9 was not just binding the CTG repeats at the DNA level and inhibiting transcription, because then that could be another, that could be interesting, but that's not what we believe was happening, because we were using these endonucleases, right? And so we were able to show that if you express this GFP, and then at the same time you transfect Cas9 GFP, but then you remove the doxycycline. So these RNAs are no longer made. They're no longer continually expressed. Whatever RNA that you have made from GFP and from the CTG repeats are already there in the cell. And yet these plasmids were able to destroy the RNA over time. So it's like, well, okay, so transcription is done. You have RNA in the cell. And then these proteins are able to still go there and bind the RNA and destroy them. And there are many ways to show this with dot blots, qPCR, and so on. Okay, so you can reduce these RNA foci to independent of transcription. So those are all just in cells you transfected repeats, right? Repeat sequences. What about in patient myoblasts? So DM1 patients, you can do punch biopsies, grow up these myoblasts, and you can do the same CAG10 complementary probe for CTG, for RNA fish, right? So you can see these dots, right, in this DM1 fibroblasts. In the non-targeting case, you can see these repeats. But in the targeting case, the foci is gone. This also works in patient myoblasts. So destroying the repeats are one thing, but it doesn't really cure the disease unless you can repair all the downstream changes that the repeats cause, right? Which is like sequestering muscleblind and then causing splicing changes. One of the key measurements we wanted to make was can you release muscleblind back to its normal duties? Can you release back to its normal localization? So if you have CUG expressed in the cell, so this is normal localization of muscleblind protein, which is nuclear, and then kind of a little bit in the cytoplasm but diffused. But if you express CTG repeats, muscleblind gets trapped. The nice thing here is that if you destroy the repeats with the CUG targeting CRISPR system, right, then muscleblind localization goes back. So that's like step 2, right? So destroy RNA foci, relieve muscle-blind localization. Now, what about the muscle-blind targets, which is downstream of muscle-blind? Okay, so I need to sort of explain this a little bit. So muscle-blind, as I mentioned, is sequestered, and then because it's sequestered, it moves away from its normal targets, which are these pre-mRNA messages in the cell to control splicing. So these are just RT-PCR images, right, in gel electrophoresis, right, where you show 2 different bands. Hopefully you can see 2 bands. These are RT-PCR primer probes on the flanking exons, right, of a target exon, which we know is alternatively spliced. So that means there are normally two isoforms of the same gene in the cell. So control myoblasts, and then DM1 patient myoblasts. Okay, so you can see there's one band here for control patients with non-targeting guide, and then targeting guide, you've got still one band. That's fine, right? Because it's not destroying any CDG repeats. But in the DM1 myoblast, you see there's a funny isoform. There are two isoforms now that's being generated because muscle-blind is sequestered. While with targeting guide, you can restore the isoform sort of back to normal, which is kind of nice, right? And you can do this not for just one gene, but here are the main ones that, for example, the FDA is using to measure efficacy for a DM treatment. But we said, let's just, what about all the other ones? So muscle-blind is a splicing protein, which means it affects not just one or a few, but hundreds of targets. So we did RNA-seq, and just to show on a few examples how they look like before we look at the transcriptome-wide view, you can see that in the myoblasts from patients, from DM1 patients, CDG targeting guides is destroying the repeats, right? So the isoform now is switching back to wild type, right? Whereas in the DM1 patients with GFP treatment or non-targeting guide, you still have these funny isoforms that are being created. So genome-wide look is as follows, right? So each dot here, each dot is the splicing inclusion of a particular exon for hundreds and hundreds of exons. And it's a measure of 0 to 1, it's 100% inclusion or exclusion. That's different between the DM1 non-targeting guide, right, and then just control non-targeting guide. So basically, it's the difference between DM1 myoblast and then control myoblast. We remove all the dots in the middle because those are splicing events which are not very different between DM1 patients and control myoblasts. So all the dots here are isoforms which are the most different. What's nice is that if you treat with CDG targeting guide, you see that you have a reversal, or basically all these dots here are like moving back to the middle. So they're becoming normalized back to a control, what it would look like in a control patient. Okay, so we see sort of a dramatic reversal of splicing changes in patient myotubes as well, right? And in treating this. So this actually is the sort of the greatest and most precise number of changes, right, to show restoration of the aberrant effects downstream of an expanded repeat that we've ever seen. So others have used oligos, siRNAs, small molecules, right? This is a sort of a dramatic shift back to normality. So this was then published showing that we think we can hit DM, Huntington's disease, and then in the paper we mentioned a little bit and show some data on C9 ALS. And so then, people are very excited, and we were again embarrassed because we said, well, we have not even tried anything in vivo. So then, this is forcing us to run faster than reporters, which is not a good thing. So we said, let's try this in vivo. And so we tried 2 experiments. One was in a DM1 mice model. So we can use AAV, and we managed to shrink Cas9. So in the original paper, we were able to cut out all the components of Cas9 which bound DNA. So we made it even smaller, able to then put the guide RNA in the same vector, and then treat mice with a single vector AAV. Now there are other forms of Cas, so different CGE Cas9, SA Cas9, which are actually smaller, and we're able to sort of engineer also smaller endonucleases, put them all together in one AAV. So in these mice models, they express long repeats, so LR, long repeats, in the muscle of these mice, and they get DM1 type phenotypes. So as an experiment, we injected the guides here, and then a CDG targeting guide, and then a control guide. and just to see whether we get molecular restoration, cell biology restoration. Okay, so what was comforting was that when we measured splicing again, so again, the few key ones just to show on by RT-PCR, we're able to see that in the mice model, you have these two bands representing different isoforms of the chloride channel, one gene that's disrupted because muscleblind is sequestered. But if you target the CDG repeats, right, RNA repeats, you're now restoring the isoforms back to normal. In fact, you restore not only the splicing, but the protein levels of the chloride channel gene, which is the key gene important for myotonia in people and obviously in the mice. So in green is the chloride channel protein levels, which is restored in the CDG targeted limb of the mice, but not in the non-targeting. And so you can restore about 86% of the splicing markers in the mice as well. Okay. Great. Well, a few more slides. And then so we also see that we can actually reduce myonuclei, which is a hallmark of sort of the disease pathology you see in the mice and also in patients. So this is now restored. I sort of want to go over it sort of too much detail. And so far we don't see a tremendous immune response, which is kind of surprising. So far. So this is still ongoing work. We're still working on different components of this. But after the second paper, folks wanted us to try to push to non-human primate models, which is the second model, and then hopefully to people. And so we have a small little company in San Diego attempting to do that. And so that's sort of ongoing. Okay, we think that this idea is kind of odd, right? Because in the space of sort of genome engineering, but it's actually a rapidly growing area of interest with papers coming out from Feng Zhang's lab, right, Jennifer's lab, and now also a company, RNA sort of company called Arbor Therapeutics, and Patrick Xu has a new also paper at the same time of a new Cas protein that binds only RNA. So I think it's an interesting area. It's growing. A lot of things you can do with this. I hope you get a chance to do something useful. All right. So just so the first paper I mentioned was the first RNA targeting of Cas in live cells with Jennifer's lab. And then a demonstration that we can do this in microcell expansion diseases as a proof of concept. This is together with Maury Swanson, who's an expert in DM, and the two postdocs in my lab, Dave and Ron, worked on this project. Dave was a grad student then, and now both Dave and Ron are full-time in Locana, trying to push this to actually hopefully people. So with that, I'm happy to take questions. Thank you very much to my lab at UCSD and for the invitation.
Questions?
Thank you for a very inspiring talk. I'm curious, and I'm sure we'll talk through this, I'm sure. Give us some sense of how you would compare this to antisense and RNAi.
Great question. So in the original manuscript for the DM cases, reviewers asked exactly the same question. Have you compared the siRNA, ASOs? So the problem with siRNAs is that they work really well in the cytoplasm. These repeats are nuclear repeats, so that was an issue that we tried and we couldn't get downregulation. ASO, so Ionis's ASO trial for DM got shut down this year because of bioavailability. So they're able to get the ASOs, as you know, they diffuse passively in tissues, but they were not able to get enough of them, right, in the right tissue, and couldn't get efficacy, right? So we compared their ASO data with our data, and in terms of the biomarkers that they measured these 5 biomarkers. We see dramatic change with our idea, right? But we also see transcriptome-wide reversal. So I think, for whatever reason, which you're not quite clear, right, the Cas and the guide seems to be much better at targeting these things. And the beauty is because we can deliver, beauty or weakness, we can deliver with AAV, we're able to now target with much greater cell type specificity. And obviously the issue is, well, can we keep packaging them smaller? There's a manufacturing issue, all this stuff, right? So we're working on that. Yeah, it's a great question. Yeah, Bruce. Do you need a mic?
Lovely data there. You said that you were surprised that there wasn't an immune response, and
Yep.
and actually,
We expect
so am I. What plans do you have in using it in people? Because it's known that people have antibodies to this bacterial protein, and
That's right.
And it's also been shown, for instance, where people take cells that express, say, the transactivator in it and transfer it to other cell types that you can have massive, detrimental immune responses to bacterial protein.
Right, great question. So within the lab, we're exploring different applications of this now, but within the company, I can say that they are evolving ways to minimize epitope presentation of the protein. We've also now done screens to find other proteins which people have not been exposed to in the space. And we are trying different regimens, so immune modulators at the same time to build tolerance to the protein. So there are efforts ongoing. I think some of these efforts are also being performed by the genome editing companies, right, editors and so on. So I think we are in good company in terms of the hurdles, I think, for immunogenicity. But yeah.
But I mean, for genome editing, you just need,
right.
I mean, they call it
hit and run, right.
Right.
Yeah.
And this is hit and stay.
That's right, yes.
And the staying part for immunogenicity is a lot.
Yes. Yeah, we are doing the in vivo models now. So far, nothing. Treating with different immune modulators, we've been actually, it's been tolerable. Let's put it that way. So still working on it. Yeah.
I wonder, in your in vivo experiment, have you demonstrated the functional the contractile force?
Right. So those are being measured. We're doing rotor-rod treatments, measurements, and now we're doing some work in the CNS for DM as well, DM mice. So there we're looking at other behavioral phenotypes, but that's still ongoing. It's been 6 months, basically, yeah.
Over here. Just repeat.
Well, so there are 150 something gene therapy trials so far in the world, and I think there are examples where transgenes have been around for 10, 15 years. So we don't know, right? And that's something that we will have to evaluate. Can we keep the transgenes in there for the lifetime of the individual? Yeah. So these are all issues that we and the other folks in the gene therapy field are exploring. Yeah.
Sorry, I have one quick question. So what about non-coding like tRNAs and rRNAs and all that?
Yeah, so we've been able, in principle, you should be able to target, circular RNAs, tRNAs, and so on. We are also evaluating that more for basic biology things in the lab. Yeah.
Great to visualize that in the nucleus and the nucleus.
Right. Yeah. Yes. Tools.
Yeah. Thank you, Gene.
Yeah.
Very interesting talk. So our last speaker